Welcome to the lesson on meiosis. So in the last chapter we learned about mitosis, and in this one we're learning about meiosis. So meiosis is the process by which we make sperm in males and eggs in females. And sperm and eggs are kind of special because unlike the other cells in our body, which have 46 chromosomes, sperm and egg have only 23. They have half the information the other cells do, so they are haploid. All right, so meiosis is the process by which a diploid cell, which we have here and here, these are called germ cells, by the way that a diploid cell, so a cell that has 46 chromosomes, goes through two divisions. So we're going to split twice. Here we're splitting once into two, and then we're splitting one more time into 4 cells to produce genetically unique haploid cells. So these sperm cells have half the information, and they are genetically unique. That means that each sperm is different from the next sperm. And here is the egg in females, and each egg that a female produces is going to be genetically different from the parent and genetically different from all the other eggs. So it's kind of special and different from the way mitosis works. So back to the human life cycle, right? We've always started here with the egg and sperm. And the egg and sperm have to be haploid. They have to have half the information because what they do when they come together in fertilization is they join their information to produce a zygote. And you need the zygote to have 46 chromosomes, so the egg and the sperm each have to have 23. 23 plus 23 will give you 46. So in the last chapter, we learned how the zygote goes through mitotic division or mitosis to produce copies of itself, genetic identicals of itself, and those copies differentiate into different types of cells like bone cells and heart cells and muscle cells and brain cells until you end up with a very complicated organism like a human. Now, this chapter, we're going to focus on this part, where in a female's ovary and in a male's testes, the process of meiosis happens, and we produce egg and sperm, which are haploid. So how do we go from 46 chromosomes down to something that has only 23? That's what meiosis does. So here is the first part of meiosis. There's 2 parts of meiosis. In meiosis 1, we're going to split a cell one time into 2 daughter cells. And in meiosis 2, we're going to split one more time so that we end up with 4 daughter cells. So in meiosis 1, homologous chromosomes are separated to form 2 daughter cells. So if you remember, we talked about this idea of homologous chromosomes. We know that not only do we have 46 chromosomes, we know those chromosomes come in homologous pairs. That means that you get one from mom, one from dad of chromosome 1. You get one from mom and one from dad out of chromosome 2. So let me show you how that looks, right? All right, so in a zygote, we have 2 copies of each chromosomes. They're in pairs. These pairs are the same size, they're the same shape, and we call them homologous pairs. All right, so that's what we're talking about here. Now, in meiosis 1, the homologous chromosomes are going to be separated. So in prophase 1, we have normal prophase stuff happen. Look, the nuclear envelope is breaking, the spindle fibers are forming, nucleolus is disappearing. So all that normal stuff is happening, but then something else is happening. The homologous pairs are finding each other. So that's one pair, that blue one from dad and that green one from mom, and that pair over here, that little blue one from dad and that little green one from mom. And of course, for us, it's not 2 pairs, it's 23 pairs that will find each other. And what they're going to do is they're going to do something called crossing over, where they're going to exchange little bits of information with each other. So between the homologous pairs, the one from mom and the one from dad will exchange pieces to get themselves really jumbled up. In metaphase 1, something super weird happens. Instead of lining up single file on the metaphase plate, the homologous pairs are going to line up with each other. And then in anaphase 1, it's the pairs that are being separated, not the chromosomes that are being, the sister chromatids that are being ripped apart. It's the pairs that are being separated. And during telophase 1, we end up with 2 daughter cells, each having these different homologous chromosomes that have been separated from each other. So we're not done here. This is meiosis 1. We're not done. We have to do meiosis 2. Meiosis 2, we're going to do another route. So we're going to do prophase 2, metaphase 2, anaphase 2, and telophase 2. So here's prophase 2. And a new spindle forms, nuclear envelope breaks down again, nucleolus disappears, all that stuff, right? And then in metaphase 2, now they're going to line up single file. The chromosomes will line up with their centromeres along the metaphase plate. And during anaphase 2, the sister chromatids will now be torn apart from each other. And in telophase 2, we're finished. We have 1, 2, 3, 4 daughter cells. And these daughter cells are haploid. They have half the information that the original parent cell had. And if you take a close look at each of them, each of these daughter cells are different. So this one you can see has like a big blue one and a little green one with a tiny piece of blue, and this one has a big green one and a small blue one. They're all different from each other. In females, this would form into actually egg cells, right? And we haven't talked about this yet, but in females, only one of them would become an egg cell and the other 3 would actually dissolve into polar bodies. And if this is a male, you're going to form 4 sperm. So this will become a sperm, this will become a sperm, this will become a sperm, and this will become a sperm. So here's a nice overview of the process of meiosis. You have a parent cell. It's going through 2 divisions. So here we're copying, actually, so first we're copying everything because we still have interphase happen. And then we have 1 division over here, and we have a second division over here. And if you look at the daughter cells, they have half the information that the parent had, which is what we expect. Okay, so here is a closer look at crossing over, which happened in prophase 1. And in crossing over, you have the homologous chromosomes exchanging pieces. So you can see this chromosome has a little bit of the blue, and this one has a little bit of the red. This is crossing over. Okay, so let's talk about gametogenesis, because meiosis happens differently in females than in males. In females, we call it oogenesis, and in males, we call it spermatogenesis. And just to see the overview here, in females, we still end up with 4 new daughter cells during meiosis, but only one of them will become an ovum, a mature egg cell that can be fertilized. The other 3 will become these things known as polar bodies that will disintegrate and die. In males, you're going to form 4 sperm. So quick information about spermatogenesis. This occurs in the testes of the male. Men produce sperm for their whole life. They produce about 250 million sperm per day, so it's a lot of sperm cells being made through a lifetime. But remember that sperm cells are very small in comparison to the egg. So here's spermatogenesis. So this is just meiosis inside of a male. So this is our diploid cell, and you end up with 4 haploid cells. Each one becomes a sperm cell. In females, this is a little bit different. Oogenesis occurs in the ovaries, and actually females are born with all the eggs they're ever going to have, but those eggs are in an immature form. Starting at puberty, females begin to ovulate, and they release 1 egg every 28 days. So here is oogenesis. So we still start with something that's diploid right here, and we finish with 4 haploid cells. But out of these 4 haploid cells, this one, and this one are going to die because they actually don't get enough cytoplasm. It's the egg that gets most of the cytoplasm. You want to think of the egg as like very important. It has to be large. It has to accept the information from the sperm. All the organelles in the egg have to be used in the zygote that could potentially form. So the egg has to be very big, very strong, very robust. So during meiosis, we only form 1 egg instead of 4. And here's a mature egg cell. Okay, so genetic variation. Meiosis and sexual reproduction lead to organisms that never existed before. So you are unique. There's nobody else like you. You're genetically diverse from all the other human beings on the planet. And there's a reason for this. There's really 3 main reasons for this. The first reason is that when we make egg and sperm, in meiosis, if you remember during prophase 1, there's something called crossing over. And in crossing over, you're going to have the chromosomes from your parents jumbled with each other to different combinations that never existed before. And we have something called independent assortment and random fertilization too, which we're going to take a look at next. So here's crossing over, that jumbling that occurs, right? So things get very mixed up when we make eggs and when we make sperm. The second thing is called independent assortment, and this can be a little bit confusing. But if you remember during metaphase 1, the chromosomes lined up with their homologous partners, right? So there's the partners lined up, here are these 2 partners lined up, and for us, there's 23 pairs of partners. Now, it's totally random how they line up. Like on this possibility, all the green ones lined up on the left side. So all the chromosomes from mom are on the left, and all the chromosomes from dad are on the right. Sometimes it doesn't happen this way. There's nothing to say it has to happen that way. You could have a green one on this side over here, so like that, and you could have a green one on the right side over here. And there's like a huge amount of variety or variability within an organism like us, who have 23 pairs. They can line up all differently, and it matters how they line up because it's going to produce different combinations when you're making egg and sperm. So you can see that these are different from these. Okay, so that is independent assortment. So during metaphase 1, pairs of chromosomes can line up in equally probable ways. And the last part is called random fertilization. So each zygote is formed by the random joining of an egg and sperm. So a female releases one egg during ovulation, and it's totally random which egg she's going to release. And remember, all her eggs are genetically different from each other. In males, there's a tremendous amount of sperm, and each sperm is different from the other. So if we take a pair of parents, there's 70 trillion different combinations that they can have. So let's say your parents decided to have another 70 trillion children. Chances are they would never produce exactly you again. So it's incredibly unlikely that you exist, and you're incredibly unique. All right, so this is called random fertilization. And together, meiosis and sexual reproduction create that tremendous diversity that you see in populations that reproduce sexually. So I hope that was helpful.